Hello, my name is Luciano Abriata. I'm going to show you work we have done with my colleague Fabio Cortés Rodriguez, who is a webXR programmer, where XR means virtual reality, augmented reality, etc. We, in our lab, develop this kind of tools to assist education and work in chemistry, biology, etc. So we work at EPFL, École Polytechnique Fédérale de Lausanne, in Switzerland. What I'm going to present here, it was done at the Laboratory for Biomolecular Modeling by my colleague Fabio and me, Luciano, funded by the Swiss National Science Foundation right now, and at the beginning, when we were testing the first prototypes, by Hassler Stiftung from Bern. So what you have here is I'm using an Oculus Quest 2 device, which has it's a device for virtual reality, which has a web browser where you can create virtual reality content for the web. Okay, the device can detect my hands. Okay, and Fabio has programmed ways to introduce virtual objects here that you can then grab literally with my hand, with one or with the other. Okay, and then by using my two hands I can zoom objects. Okay, let's leave this where it belongs, here. And what we have set up in this virtual reality scene is a series of 3D models that cover topics about chemistry and biology. Okay, the idea of the presentation that we prepared in VR is to have multiple people seeing, interacting with the objects, teaching and learning in virtual reality. So here it's only me, but we could have here more people and we would all be sharing the same virtual reality session. This is part of our vision about how science will be taught and done in the metaverse. So by using highly fluent human-computer interfaces like this, where you can literally grab and move objects with your hands. And so let me move with you quickly, let me guide you quickly through this presentation that we have here. We start with a model of the human circulatory system. You see in red and blue all the veins and arteries of this 3D model of the circulatory system. You see the heart and the kidneys around here. Okay, this would be a very tall person. You see the high density of arteries and veins around the heart and around the kidneys, and then you see the thick veins and arteries that distribute the blood to the main through the main pathways, and then you see the thinner capillaries through which the blood moves. Then here we have a 3D model of a heart that we could also grab and move around. But I want to stress that we have there a circulatory system. So it's a system that is made of organs. The heart would be an organ, part of this system. And then organs are made of tissues, okay? And here we have a piece of tissue. This is actually not part of the heart. This is a part of dermal tissue. You can see you have epidermis, dermis, then the subcutis. You have these structures from which hairs go out, and then in yellow it's a kind of very fatty tissue under the dermis. Then tissue is made of cells, okay? Cells can be of various types, eukaryotic, prokaryotic, and then even us within our cell, every piece of tissue has multiple different types of cells, okay? So here we have a prototypical cell from an animal. What do we have here? So this would be very, very small compared to the tissue. It's like we are zooming in as we move from the system to the cells. Cells are like the minimal unit of life. They consist in a membrane, here called the plasma membrane, that contains all the machineries that allow the cell to grow. Okay, we have, for example, here this kind of rigid strings that form what is called the cytoskeleton, is what gives the cell some shape. Then we have the membrane that contains this is like the main limit between the interior and the exterior of the cell. And then inside the cell we have organelles, like you see here, the mitochondrion, which has two membranes in itself. We have some vesicles that can shuttle things from the outside to the inside and then to other membranous systems. This is the endoplasmic reticulum, which is a membranous continuation of the nucleus. These red dots are ribosomes, which synthesize proteins. This is the Golgi apparatus, which has a series of other functions. All of these are also membranes, okay? And you can see some vesicles that bud out of the Golgi apparatus and go to the membrane. And then in the nucleus, this main part here, we have the genetic information, which is in the form of these blue things you see here, which are chromosomes. Okay, these chromosomes, they actually consist of compacted DNA. This is compacted DNA that I could maybe here grab and pull and uncompact, and that's what we have here. This blue thing would represent one of those chromosomes. We are zooming into it. You can see how this is very compact, but we open it slowly. And then by here, what we have is that DNA, which is this blue thing, is wrapped around these proteins, which are called histones. Okay? And then we can open it more and more, and each of these sticks represents a base. So this A, C, G, T that you may have seen if you heard about DNA. And so far from the circulatory system up to here, what we are seeing are somehow 3D drawings, we could say, okay? However, the work that we do in the lab, and many other people around the world, consists in understanding each of these molecules really at atomic level, not just with drawings, but really trying to see the atoms. So that's what we have, for example, here. Here you have a 3D drawing of a nucleosome with 8 proteins and 2 rounds of DNA around. But how do we know this? Well, we know this because scientists have done experiments where they have crystallized the proteins. So you have 8 things here, and you have 8 helical things here, which are the proteins, okay? And then you see we have around these 8 proteins, we have 2 turns of DNA. I can move around so that you can see them well. It starts here, it's coming here, and then it goes on the other side, and then it's coming here. This is coming from a real experiment that we will see soon. Of course, molecules are made of atoms, and we are not seeing any atoms here. This is because we are using a very simplified representation of what a protein and what DNA are. But before I explain you how we know these structures, let me show you another example here. So we keep opening up the DNA, and then when we are here, we can again look at an actual X-ray structure of a piece of DNA, which is this. You see you have 2 backbones that are going around, and then these things are bases. And you can see that the bases are stuck in front of the other, okay? They are stacked vertically, let's say, if we consider the DNA like this. They are stuck vertically, and also they are interacting with each other, okay? Each of these is what can be an A, C, G, or T. You see, the backbone is always the same, but the thing inside, the base, is different. And what we have special here is, so you can recognize that there is this groove, quite big, then this groove that is smaller, okay? And what we have on this side, here in this case we are seeing the atoms, so each of these ends of the sticks are atoms. What we have on this side is a protein called the transcription factor, which binds to DNA in order to read the information. So you can see you have the major group here or here, and then you have the minor group in between. You can see how this protein is docking here and establishing interactions directly with the bases. This is how it can read the information, okay? Another thing we can see is that, so these are usually positive atoms. These are negatively charged groups. Then you have the same here, a positively charged group with a negatively charged group. You see how this allows the protein, the transcription factor, to bind to the protein. And then these other groups inside the DNA, we can make this even bigger, so we can go inside. You can see how these are then reading the information from the bases by doing here what is called the hydrogen bond, and then also another one here, for example. Okay, so now this again is an actual X-ray structure determined through a technique that is called X-ray diffraction. What is X-ray diffraction is one of the techniques that we can use in the lab to get to know the structures of proteins. So what you have here is a piece of a protein that has been crystallized, okay, and through which researchers shoot X-rays that then diffract. What they collect, what the scientists collect, is actually the diffraction pattern. And from the diffraction pattern, they reconstruct this white thing, which is the density of electrons, okay? And then, through a few cycles of iterations, they place atoms inside. And the kind of things we can see here is you can fit the atoms in the electron density, and in this way you get to know the structure of the protein. And interesting things is, for example, here, this that you have here is a water molecule. So you only see an oxygen atom because hydrogen atoms are too light, so they are not seen in this kind of experiment, but they are there, of course. Everything that you see here in cyan is carbons, blue is nitrogen, red is oxygen, so you're not seeing any hydrogen, but all these atoms are connected to hydrogens as well. Anyway, that we know it from the basic chemical principles. So if we see the carbon, the nitrogen, the oxygen, then we can model the rest. Here you can see when the resolution of the information is big enough, you can even see the holes in this kind of ring structures that we have. And then these waters are important because they are bound tightly to the protein. That's why we see them. Usually water will be moving around, but really close to the surface of a protein, they can stay still, okay? And normally, or very often, waters that are associated with the protein, they have a functional role. This is another kind of experiment that we can do also to try to understand proteins where the resolution is very low. We cannot place atoms here, but sometimes we know already what shape the molecule can have, and then we can place it there. Why is it important to know the structures of proteins? Well, first, because we can understand how they work, how proteins bind to DNA, for example, to read the genetic information. And another reason, a very important reason, is because if we know the structure of a protein, we can also design drugs that will modulate the activity of the protein. For example, here what we have is an enzyme called human carbonic anhydrase. It's an enzyme that we have that helps to regulate the pH of the cell, okay? Now, again, here we are not seeing all the atoms. Most of the protein is kept with a very simple representation, but we are seeing the atoms, and in this case we do see the hydrogen atoms of all these atoms here close to what is called the active site of the enzyme. The active site is where the chemistry happens. So what we see, this enzyme is special in that it has a zinc ion at the center. You have these, and these are three kinds of amino acids, parts of the protein, that can bind the zinc ion, okay? And then what you can do is, suppose you have to modulate the activity of this enzyme in order to cure a disease or to exert some other effect, then you can take a drug like this, which is a compound that will come here and it will like the shape of the protein, and then it will bind here. And when it binds here, it blocks the activity of the enzyme, because then the natural substrate cannot bind anymore, okay? If you know a bit about chemistry, you will see again that you have these nitrogens coordinating the zinc, okay? And then also this compound that we added, this drug, also has a nitrogen that can coordinate there. Likewise, you see here some very hydrophobic groups And here, indeed, the compound has a methyl group that will like to bind to these hydrophobic groups. So let me zoom this out, leave it back there, and let's go back to our cells. In this case, we will look at a plant cell. So we have a plant here. If we could zoom enough, we could start to see one of its cells. So just like humans, plants are eukaryotes, but their cells are quite different. They have the main things, like you see, you have mitochondria, you have then a nucleus with chromosomes, which will be similar to that. But here, plants have a special organelle. You also have the Golgi apparatus, but plants have a special organelle called the chloroplasts, which is green because it has a lot of chlorophyll, a green pigment. In chloroplasts, it's where photosynthesis happens and where light from the sun is used to oxidize water and produce oxygen, okay? So the oxygen that you produce, that the plants produce, it comes out, it diffuses, it goes out of the plant, and eventually it is breathed by an animal like us, and it will go through the capillaries, it will reach a cell, and in the cell it will reach a mitochondria, okay? Mitochondria have two membranes, as I told you earlier, and you see the inner membrane, the one that is very rough. The inner membrane has a very special protein called cytochrome C oxidase. It's this protein here. Again, this is not a model, but just like this, it's a protein whose structure we know through X-ray diffraction, okay? I will leave the oxygen here for a while, and I wanted to show you. So this enzyme has a quite complex structure, and inside of it, it has these and these, which are called heme groups. Then this has these two copper ions very close to each other that confer this a purple color. These groups have the typical color of blood, so very dark red. This takes electrons and shuttles them to the heme groups, and then one of the heme groups has an iron ion which has some electron density going out of the plane, you see? Now this electron out of the plane can interact with this, which is called a molecular orbital of oxygen. So oxygen eventually, when it's inside the cell, it goes to the mitochondria, and eventually it can diffuse into the protein, and by overlapping its orbitals with the orbitals of the iron ion, then it can bind there. Then it will receive the electrons coming through here, and oxygen will be reduced to water. So you have a kind of gaseous molecules coming in and then water coming out. That final delivery of electrons from the protein, the cytochrome C oxidase, to the oxygen to create water is the end of the respiratory process, which is very complex, but is essential for our life because this is the way how we produce energy. We can talk about that eventually separately. Now, to look at one more thing, I showed you at the beginning that I have here a model of a virus. In this case, it's a SARS-CoV-2 virus. This is a drawing, but it's quite accurate. So this virus has a membrane, and then it has some proteins here, the spike proteins that you may have heard, which can bind to some receptors in the cells in order to go in. Now, if we zoom enough here, we start to see inside of the virus, you have all this red thing which represents the nucleic acid. So it's the genome of the virus. So essentially, what the virus does is it enters somehow into the cell, into the system, and then it will diffuse, and eventually these pink little proteins called spikes will find a receptor on the surface of the cell. The receptor is not drawn here, but the surface of the cell is full with different kinds of proteins. Some of them may be hijacked by the virus that will then inject inside its genome to use the protein machinery, the ribosomes of the cell, to create new viruses. Okay, and then eventually they will form these kind of things and go out in millions, and then they will go and attack other cells. So with this, I have finished the story. There's much more I could tell you. For example, we have models for other systems. There are websites where people create and deposit models of different things. For example, this model of the chromosome unrolled until we see the basis was done by a digital artist, and we bought it from them. Then proteins and DNA like this and that, or like that protein there, or that other one there. There are hundreds of thousands of systems whose structures we know experimentally. So there's a lot of, there are many systems described at atomic level that we could eventually place here. In the same way, you can create your own content. And right now, this website is not open, but it will be open soon with some predefined content that you can go and use directly. And again, I tell you, here I'm only one person, but in principle, the app can support easily 5-6 people, probably even more, using this VR headset to grab molecules, or also using a telephone, a smartphone, or a tablet, or a normal computer, where of course you cannot grab the things, but because the app is a web app, so it's a web page, you can just access it from any of these devices, and at least you can see what is being explained. So with that, I will go out of this virtual reality session just to show you another thing. Welcome back. I'm here in a prototype where we don't have all these static virtual objects, but on the contrary, we have molecules which are described atom by atom, okay? And as you see, these molecules are moving. This motion simulates the effect of thermal fluctuations, so fluctuations due to temperature. If the temperature were 0, the molecules would be standing quiet, but because the temperature in this kind of simulation is not 0, then the molecules are moving. And then what we can do here is to grab an atom and move the molecule around, okay? In fact, I can grab it on 2 places, and indeed, there could also be another person, and we could interact with 4 hands at the same time on the same molecules, okay? The goal of allowing you to move the atoms by hand is that you can explore the shapes of the molecules, maybe for education or for teaching, and why not also in the future also to assist researchers. Let's see some examples. Here we have this molecule is called butane. Okay, you can recognize that you have 1, 2, 3, 4 carbon atoms, and then butane is a very flexible molecule. I can stand here and I can rotate the atoms in the back, so I can put these 2 groups on the same side, or I can put them on opposite sides. Of course, with temperature fluctuations, you see this moving now, and if you wait long enough, eventually these rotations will happen naturally. Actually, it's starting to happen there. Now, I wanted to compare butane with this molecule, which also has 4 carbon atoms, but it has a double bond in the middle. You see this bond is thicker than the others. Double bonds are non-rotatable, okay? And indeed, if I try to rotate that, so let's try to do the same I did before. I will put these 2 carbon atoms there, and then I will say, okay, now I want to do this rotation. You see, it tries to do it, but it cannot, and eventually it comes back. I can try to apply a lot of force, maybe like this, but still, I cannot. Okay? This is because the double bond, it's rigid, so the atoms stay on that plane and they cannot go out. And here we have another molecule, very interesting, especially to look at it in 3D. This is called adamantane, and you can see there are 10 carbons only, but they are fused in a way that they make up 3 rings. You see one ring here, the rings share some carbon atoms, one ring here, another ring here, and then the other one is here, okay? So the molecule, because of the shape it has, and because it's 3 rings fused together, is very rigid. This you can compare to any of the other molecules which are moving quite a lot. Then let's see some other molecule, maybe with some more colors. This one, which I grabbed at the beginning, is an amino acid called methionine. Amino acids have nitrogen, carbon, a carbon called alpha carbon, and the carboxylate here. And then, this is always the same for all the amino acids. And then they have a side chain that is different. For example, here we have methionine, as I told you. This is the yellow atom is sulfur. And let's say here I have another amino acid called arginine. You can see again that you have nitrogen, carbon, carboxylate, and then a side chain, which in this case is different. Arginine has, at the end, instead of having a sulfur with a methyl group, this has a group called the guanidinium group, which, as you can see, is quite planar because you have also double bonds in there. Let's take another example, this one. This is yet another amino acid. Again, you have nitrogen, alpha carbon, carboxylate, but this amino acid has 2 aromatic rings fused. So this one that has a nitrogen here, and then this one has 5 members, 5 atoms in the ring, and then this one with 6 atoms. You can see that because it's 2 fused aromatic rings, then the whole thing is very aromatic and very planar. So I can try to deform it, but I cannot. Well, this virtual reality app I'm showing you now, in principle, can load any small molecule. So in the future, I think students and teachers will be able to explore chemistry very interactively, especially everything that has to do with conformations, just by grabbing molecules and moving them around as if this were a plastic modeling kit, but in virtual reality, which has many advantages. For example, you don't have to build the molecules, you can have many more atoms, you can have complicated molecules, you can simulate realistic motions, and this is not supported now, but in principle, one could also simulate or emulate chemical reactions, for example. And I repeat, here this application now supports 2 people working together. In the future, we have to rewrite it to allow more people to interact, but this is what we are starting to do now in the lab. And again, same as the other thing I showed you earlier, this is all web, which means that you can go inside virtual reality with the web browser that you have in the virtual reality headset, and then other people can follow the session remotely by accessing the same web page on their, say, computers or smartphones or tablets. And the experience is, of course, not so immersive, but at least it is there. With that, I say goodbye, and well, hopefully in a couple of months we will have another video showing you more content, and also hopefully giving you the web address to go use these applications yourself.